After the mechanical energy of sound has been transduced into neuronal activity in the inner ear, it begins its journey to the brain. Signals from the hair cells in each ear's cochlea are sent through the spiral ganglion cells and auditory nerves to a sequence of nuclei in the brainstem, before eventually reaching the midbrain, thalamus, and the auditory cortex. A complex of nuclei in the brainstem, known as the cochlear nucleus complex, cochlear nucleus, or simply CN, is located in the dorsolateral side of the brainstem, spanning the junction of the pons and the medulla. The CN consists of two heterogeneous collections of neurons whose inputs come from the cochlear nerve, and whose outputs go towards higher regions of the auditory brainstem. Auditory nerves innervate the cochlear nucleus in a spatial pattern that matches their spatial organization in the basilar membrane of the cochlea, forming a highly organized network based on the frequencies to which each nerve is tuned. Auditory nerves carrying signals from hair cells near the apical basilar membrane have low characteristic frequencies. Those from hair cells near the basal basilar membrane, near the round and oval windows, have a high characteristic frequency, and so on. When these auditory axons innervate the cochlear nucleus, they do so in a highly organized pattern based on their characteristic frequencies. This systematic relationship between the position in the cochlear nucleus and characteristic frequency is another example of tonotopy, a topographic organization of neural responses according to frequency. Tonotopic maps are found throughout the auditory system, beginning at the basilar membrane and carried forward in each subsequent processing stage, just the same way that many structures in the visual system carry retinotopic organization all the way to cortex. After the cochlear nucleus, auditory signals are carried deeper into the brain via two distinct pathways before ultimately reaching the auditory cortex. One set of signals is sent from the cochlear nucleus to the superior olive. The superior olive marks the first place where auditory signals from both ears come together, are compared, and integrated. Two specialized groups of cells in distinct regions of the superior olive encode differences in sound intensity, or loudness, as well as timing differences between the sound perceived by the left ear versus the right ear. From the superior olive, auditory information is sent to the inferior colliculus, which is the principal nucleus of the auditory pathway in the midbrain. Anatomically, it sits below the superior colliculus, which is also known as the optic tectum, which we learned about in the last lesson. The superior colliculus plays an important role in directing and orienting head and eye movements in response to visual stimuli. Since these two structures are so close together, it's perhaps not surprising that the inferior colliculus serves an analogous coordinating function, allowing us to orient ourselves in response to auditory stimuli. The second set of signals leaving the cochlear nucleus bypasses the superior olive and goes straight to the inferior colliculus. Once these two sets of signals are processed in the inferior colliculus, they are sent on to the medial geniculate nucleus. The medial geniculate nucleus, also known as the MGN, is the auditory subdivision of the thalamus, and it basically is to the auditory system what the lateral geniculate nucleus, or LGN, is to the visual system. It's made up of a number of distinct cell types, forming distinct layers, and carrying out specific functions to help us hear. It's also thought that the MGN plays a role in allowing us to direct and maintain our attention to a particular sound.